The nervous system is divided into categories based on both function and structure. Think about how we did this in Chapter 9 with joints. We went through all the joints and classified them by their structure, then we classified them by their function. So we're going to do the same thing with the nervous system. Over here is structure, and this side is function. For structure, we have two. There is the CNS, which is central nervous system. This is the brain and spine. The central nervous system receives information, makes decisions, and gives commands. Then we also have the PNS, which stands for peripheral nervous system. This is all the nerves out in your body. They connect the CNS to your body parts. And nerves have structures on them called ganglia. Ganglia are swollen areas of nerves, kind of like a little knob on the nerve, where cell bodies are housed. And in the next section, we'll look at the parts of a nerve and you'll see what a cell body is. Then if we go to function, function has two main parts and then each of those has a subpart. First is sensory. Sensory is information that goes to the CNS, and it goes from a receptor. So sensory is information from a receptor to the CNS. The other side is motor. Motor is commands from the CNS to an effector. Effector means the body part that you're giving the command to. Okay, think of sensory and motor like a divided highway. In a highway, you have northbound lanes and you have southbound lanes. Sensory is always going to be moving toward the CNS. So it's like your northbound lanes. Motor is always moving away from the CNS. So it is like your southbound lanes. Each of these is divided into two parts. Sensory has somatic and visceral. Okay, somatic is the stuff that you know you know. It is conscious. You have conscious awareness. Visceral is subconscious. It's the stuff you don't know you know. Okay, for example, the stuff you know would be like seeing. You know what you see. Hearing, you can tell me what you hear. Smell, taste, and touch. Those are all things you are consciously aware of. Subconscious would be, for example, what's your blood pressure right now? There is a part of your brain that knows what your blood pressure is right now, but it's a subconscious part of your brain. So you know it, but you don't realize you know it. Another example would be how much glucose is in your blood right now. If you wanted to consciously know this, you would have to get a glucose meter and prick your finger and get some blood and have it tested that way. But the subconscious parts of your brain already know how much glucose you have. So visceral kind of falls into two categories. Composition of your blood, right? Like the osmolarity, the pH, temperature, and information from your organs. So like your liver is talking to your brain right now. You just don't consciously know it. Then with motor, it also has somatic, and the other division is autonomic. Somatic is voluntary. You get to consciously choose what you do with somatic. Autonomic is involuntary. It happens whether you like it or not. There's only one effector in your whole body that is voluntary, and that is skeletal muscle. Involuntary, or autonomic, has three types of effectors. As you can see in the picture there, the heart, so cardiac muscle. And to put a little note here with cardiac muscle, your brain doesn't tell your heart to beat. It beats on its own, but your brain can tell your heart to go faster or slower. Smooth muscle. Examples of smooth muscle include your blood vessels and your digestive system. So when your blood vessels dilate or constrict, depending on your temperature, you don't consciously do that. It happens automatically. Or after you eat lunch, you don't have to consciously sit there and digest your lunch. That's all autonomic. And the third effector is glands. Think about when you sweat. You don't choose to sweat, it just happens. Or salivary glands, when your mouth waters. Those are all autonomic activities. So there are the divisions, the classification of the nervous system. Now we are ready to look at a neuron and its parts and see the beginnings of the functions of how a neuron works. So in this picture is a neuron, and a neuron is a single cell of the nervous system. And this cell conducts electric signals. Now think back to chapter 2 for a few minutes. Remember when you mixed salt and water and you had the buzzer and you were putting the probes in to see if it would conduct electricity and make the buzzer loud? You use ions to conduct electricity. That's what your neuron is doing also. And do you remember from chapter 12 or chapter 2 where you get ions from? They're from electrolytes, which are in your food. So food and drink have electrolytes. Those electrolytes mix with the water in your body. They dissolve and dissociate and produce ions. Ions then conduct electricity. Go back to your chapter 2 lab for a review of all that. So the neuron conducts electric signal by moving ions in and out of the neuron. And this signal is one way. So it enters through this part of the neuron, it moves through like this, and then you get down here, and that's where you then transmit the signal to something else. Over on the right, it's color-coded by region. So we have the blue, the pink, the green, and the purple show us the different segments of a neuron. So we're going to go through and look at each of those. The top one here is the receptive segment. So it receives the signal from another neuron. And that signal is in the form of neurotransmitter. And there are two parts of a neuron that are in the receptive segment. The dendrites, which are kind of all these things up here, all these branches are dendrites. And then this whole big round part is the cell body. And the cell body can also be called soma. The next part of the neuron is the initial segment. It initiates a signal, which is called an action potential. And this is kind of this triangle area right here called the hillock. Okay, think about when you hear gossip. Somebody tells you a rumor, so you receive that information. So your ears are like the receptive segment. Then you have to decide if you want to repeat that rumor to someone else and spread the gossip. That's what the hillock does. So signals are coming in to the dendrites and the cell body, and the hillock has to decide if it's going to repeat that signal through this neuron and send it on to the next neuron. The green area here represents the conductive segment. If the hillock does decide that it's going to repeat that information and send it on, the conductive segment is what conducts the action potential. And the conductive segment can be very long. You have the longest one would be in your sciatic nerve that goes from your lower back through your hips, down your leg, all the way to your foot. So the long part of a neuron is its axon. So all of this from here down to here is all axon. Then when you get to the bottom and it branches out, these branches are telodendria. Then the last part here in the purple is our transmissive segment. It transmits the signal to either another neuron or the effector. And it does so by secreting neurotransmitter. and it has these little bulbs here. These are synaptic bulbs. They can also be called synaptic knobs, or sometimes they're even called axon knobs. Those are the parts that secrete neurotransmitter. Okay, so those are the parts of the neuron. But remember we said that a neuron moves ions in and out. So now think back to chapter 4. What moves ions in and out of a cell? Do you remember learning about transport proteins? And we had channels, gates, and pumps. So now we're going to have all these transport proteins that we're going to label. And they're color-coded based on what goes through them. So first we're going to look at the ones up here in the receptive segment. These are ligand regulated. And remember, a channel is open all the time. A gate can open or close. See this little flap right there? That tells you it's a gate because it has a little flap that can close. So these are ligand regulated gates. Ligand means that they open when something binds to them. This little green triangle here, that is the neurotransmitter. Neurotransmitters and hormones are ligands. So ligand regulated gates open when a neurotransmitter binds to them. And we have 3 different ligand regulated gates here. The pink represents sodium. And when it opens, sodium moves in. The blue represents potassium, and it lets potassium out. The green is chloride, and it lets chloride in. Then if we move down here to the initial and conductive segments, you have the exact same gates in both of these segments, so we'll just talk about them together. So notice there's that little flap telling you it's a gate, but you don't see a place here for neurotransmitter to bind. So these are not ligand gates. These are voltage regulated gates. So they open when the voltage of the neuron increases. Okay, so neurons, they run on electricity, so they have voltage. Think of like a 9-volt battery, right? So there's voltage in the neurons, and that voltage can go up or down. When the voltage goes up, that's what makes these gates open. And we're going to follow the same color coding. So with the pink ones, these are sodium, and when they open, sodium comes in. The blue ones are potassium, and when they open, potassium goes out. Okay. Over here to the left, we have 3 more transport proteins. Look at the color coding scheme of the whole neuron here. On the left, it's yellow, right? From here all the way down to here. So what that's representing is that these 3 gates are found throughout the neuron. So the gates that are on the right in the specific sections are only found in that section of the neuron. The ones on the left are found throughout the entire neuron. So these 3 are in the entire neuron. Whereas, for example, the ligand-regulated gates are found only in the receptive segment and nowhere else in the neuron. The first one here is a sodium-potassium pump. If you look back at your chapter 4 notes, when we were learning about active transport, I mentioned this as an example, the sodium-potassium pump in neurons. Now remember, pumps use ATP, and they are going to force molecules to go where they don't want to go. So they're going to go against the concentration gradient. They're going to concentrate the ions. So this pump moves both sodium and potassium. It pumps sodium out, and it pumps potassium in. The other two are leak channels. So notice there's no flap there. They don't close. So these are channels. And you have the pink one is sodium and the blue one is potassium. So a channel is always open, but notice it says leak channel. So they let small amounts of sodium and potassium leak through. And they leak through according to their electrochemical gradient. Then if we go down to the transmissive segment, here is where you move calcium in and out. This one is a calcium gate, and when it opens, it lets calcium in. And this one is a calcium pump, and it pumps calcium back out. And this gate is voltage regulated. So notice the pump moves calcium the opposite direction of the gate. If we look back up here, the same thing. Our gates over on the right let sodium in, our pump pumps the sodium out. And our gates let potassium out, the pump pumps sodium in. So the pumps do the opposite of the gates. Okay, so this is your introduction to a neuron. It's the parts of the neuron and the transport proteins that are moving your ions in and out. In the next section, we're going to see how this works to actually make that electric signal. Now that you know the parts of a neuron, let's start looking at potentials. First, we will look at resting membrane potential. Resting membrane potential is the voltage of the inside of the neuron when it is at rest, not sending a signal. We calculate this voltage by comparing the inside and the outside. So notice there are ions both inside and outside. Here we have the sodiums, here we have potassiums. Inside are most of your potassiums. Remember that most of the sodium's outside, most of the potassium is inside. And there's some other things like this is a protein. There's other ions here also. The point is you have ions on the inside and you have ions on the outside. Each ion has a charge. You add up the total charges for the outside. You add up the total charges for the inside. Then you compare these two totals to each other. You assign a number to the inside based on this comparison. Here is an animation to help you understand this idea. For resting membrane potential, you compare how many cations are inside to how many cations are outside the neuron. Then you assign a voltage to the inside of the neuron based on this comparison. Here, Billy is inside and he has 4 apples. Timmy is outside and he has 3 apples. When we compare these, Billy has 1 more than Timmy, so we assign Billy a voltage of +1. Now Billy gives Timmy 2 of his apples. That leaves Billy with 2 and Timmy has 5. When you compare them, Billy has 3 fewer apples than Timmy, so we assign Billy a voltage of -3. Then if Timmy gives those 2 apples back, plus 2 more, then Billy has a total of 6 apples and Timmy has 1. This means Billy has 5 more apples than Timmy, so we give Billy a voltage of +5. In a real neuron, the voltage of the inside at rest is -75. And that is millivolts is our unit. So if you look here, we do have cations on the inside. You have all that positive potassium, but you have more cations on the outside. So even though the inside has cations, it's -75 because it has 75 fewer than the outside does. And that's how you calculate the voltages for the neuron. You compare the inside to the outside, and the difference between them, and it can be positive or negative, is the voltage you assign to the inside. Now that you know about resting membrane potential, let's see how we have a signal in a neuron. First, we're just going to focus on the axon, so the conductive segment. So from here to here. So we're starting off with the whole neuron in resting membrane potential. Now remember, a signal starts up here at the dendrites and cell body, and it moves through the axon and then down to the telodendria, and then this last part is the synaptic knob. We eventually will learn what happens in each of these parts. But right now we're going to see what happens in the axon. So at the start, at resting membrane potential, you are at -75 for the inside of the axon. And you have most of your sodium outside, and you have most of your potassium inside. In order to have that action potential on the axon, you have to move the ions in and out. So sodium will move in, and potassium will move out. And that's what makes the electric signal. This picture is just showing again, sodium goes in and potassium goes out. And when sodium goes in, then potassium goes out, you have a signal. Here we can look at it in more detail, and we can split it up into 6 steps of what happens in order to get a signal going through your axon. We're going to start off, here's our voltages. So this is -75, our resting. The dotted line is -55, which is threshold. Up here is +30. So those are the 3 voltages we're going to deal with. So we begin step 1. You're at resting membrane potential. And at this time, we know that potassium is in and sodium is out. Step 2 is this part right here. Let's go ahead, we'll color code that. There's step 1, here's step 2. Step 2 takes place in the hillock. This is when the hillock is doing summation. So the voltage is raising in the hillock. If it raises to at least -55, it will cause an action potential in the axon. So remember we said the hillock is the initial segment. It's like when you hear a rumor and then you have to decide if you want to pass that rumor on. So right here is the hillock deciding to do that. Then step 3 is depolarization. So the voltage is going up. So this is when the sodium gates open. And when they do, sodium comes in. Now you have potassium in, and you also have sodium in. So because both of your positive ions are inside, this raises the voltage to +30. Next is depolarization. And this time, the voltage is going down. I'm sorry, repolarization. So now the potassium gates open. And potassium goes out. So now we have our sodium is in and our potassium is out. So because the potassium is going out, a positive ion is leaving, the voltage goes down. The next part here, see we have this dip kind of below your resting membrane potential. That is hyperpolarization. Okay, remember that hyper means more. So this means that it is more negative. So think about how you would get it more negative. We got it to go negative by letting a positive ion out, the potassium. You get it more negative by letting extra potassium out. So during this time, the potassium gates remain open and extra potassium goes out. So now we have our sodium in, we have our potassium out, and we have extra potassium out, which makes it extra negative. Then the last part here, we return to resting membrane potential. Remember the pump and the leak channels? This is where they come in. They put everything back to their starting positions. And your voltage returns to negative 75. So now we have our sodium back out, and we have our potassium back in, and we're back at negative 75. So there's an overview of how you have that action potential in the axon. And steps 3 and 4, depolarization and repolarization, these two steps together are considered an action potential. So whenever you're talking about action potential, that's what you're talking about, depolarization and repolarization. And that is the signal.
Billy and Timmy are back with their apples, and this time they have some friends here too. Billy has an apple and he wants to get it to Timmy, so he passes it down the line of his friends. One by one, they pass the apple to Timmy. Passing the apple one by one down the line gets it to Timmy, but it's a slow way to do it. Billy wants a faster way to get the apple to Timmy. So he throws the apple to Janelle, who then throws it to Timmy. They skip parts of the line. This goes a lot faster than passing it one by one.
So Billy and Timmy and their friends are like an axon, and the apple represents the action potential. In the picture on the left, we have continuous conduction. This is when they pass the apple down the line one by one. So remember that the gates in the axon are voltage regulated. So a gate opens when the voltage nearby goes up to at least negative 55. In continuous conduction, you move down the entire axon, raising the voltage bit by bit. So right here, we have, you can see the sodium going in, the potassium going out. We're having an action potential. When the sodium comes in, the voltage raises. So then that stimulates an action potential in the very next section of the neuron, which stimulates an action potential in the very next section. So you're going through, you're moving the action potential this way, and if you line these up, you can see that it's just moving bit by bit down the axon. This will move the action potential down the axon, but this is a slow way to do it. Most of your neurons have myelin. And if they have myelin, they can have saltatory conduction. Saltatory means jump. Salt means jump. So in this case, the action potential jumps along the axon. You have cells that wrap around the axon. They can be called Schwann cells or neurolimocytes. So these are cells that wrap around the axon. There's gaps between the cells. These gaps between them are the nodes. They can be called nodes of Ranvier, or they can be called neurofibril node. Wherever the Schwann cell is, it's covering up the axon. So you can't have ions move in and out. It's only in the nodes where the ions are able to get in and out. So this means you can only have action potentials in the nodes. So if we look at this, here we have an action potential. You can see the sodium going in, the potassium going out. Once the sodium is in, it diffuses. So this part shows sodium diffusing. So sodium will diffuse under the Schwann cell. It then gets over here and it raises the voltage. And as long as it raises it to at least -55, then we have an action potential in the next node. And again, the sodium comes in and it diffuses under the cell. That increases the voltage of the next node, and it has an action potential. So we're still moving the action potential this direction, but we're skipping large sections of the axon. You only have the action potential in small areas, the nodes. So this was like when Timmy threw the apple to Janelle, or I'm sorry, Billy threw the apple to Janelle, and then she threw it to Billy. So the action potential jumps from node to node. And this is much faster. So to sum up saltatory conduction, you have an action potential in a node. Sodium comes in and it diffuses under the cell, that Schwann cell or neurolimocyte, to the next node, where it raises the voltage and stimulates those gates to open. And then an action potential occurs in that node. So it's called saltatory. Salt means jump because it appears that the action potential jumps node to node. And this is by far the much faster way to send an action potential down an axon. There is a disease in which you lose the ability to do saltatory conduction. So here you can see the normal nerve has its myelin, which remember is composed of those Schwann cells or neurolimocytes. In the bottom one, you can see that you're missing that. The cells have been destroyed. And so this axon loses its myelin. so it can no longer do saltatory conduction. And so transmitting action potentials becomes very slow. And this disease is multiple sclerosis. Or called MS for short. This is an autoimmune disease. So autoimmune is when your immune system messes up and attacks your own cells. Your immune system is only supposed to attack pathogens that are making you sick. But in autoimmune, it messes up and it attacks your own cells. So it's the immune system that is destroying these cells. And that's why one of the symptoms of MS is very slow movement, because it takes a long time to get that action potential down the motor neurons and secrete the acetylcholine onto the muscles. So the responses are very slow. So remember how we've talked about the action potential, the signal moves one direction through a neuron. It comes in at the dendrites and the cell body, it goes through the axon, down the telodendria, and then to the synaptic knobs, always in that direction. Well, now we're going to look at how you keep it going in that direction. And the key to that is refractory period. Refractory period is what keeps the signal moving through the neuron in the right direction. Right here, step 5, while you are hyperpolarized, is when the refractory period occurs. During this time, when it is hyperpolarized, you can't open sodium gates. And so if you can't open sodium gates, you can't depolarize and have an action potential. Once you return to resting membrane potential, so right here, that return to resting membrane potential, then you can open sodium gates and you are ready to have another action potential. So refractory period is the time when the neuron is extra negative, hyperpolarized, and that extra negative charge locks the sodium gates closed. Sodium gates can't be opened if the neuron is too negative. So let's look at how that works in the neuron to keep the signal moving in the right direction. Suppose here's an axon. And remember, the axon has its myelin. And we have action potentials in the nodes. So we're going to say here is node 1, node 2, node 3, and node 4. So node 1 has an action potential. So we're going to have an action potential here in node 1. So this means we have sodium in, and we go to positive 30, then the sodium diffuses through here, and that raises the voltage here to negative 55. And that negative 55 is then what makes these sodium gates open, and sodium comes in, which takes this to positive 30, and then sodium diffuses. Meanwhile, while the sodium is diffusing over and your next one, node 2, is having an action potential, back here in node 1, we have potassium going out, and we go to negative 75, then extra potassium goes out. Remember the hyperpolarize. While we are hyperpolarized now, this is in refractory. And the sodium gates won't open. Okay, so while node 2 is at positive 30, and that sodium is diffusing, so sodium diffusion occurs in all directions. So while node 2 is at positive 30, sodium is going to diffuse back this way also. And it's going to try to come back and raise the voltage in node 1. Think of a lamp. When you turn on a lamp, light spreads out from the lamp in all directions. But only node 3 is going to have an action potential next. So the sodium is fusing out in all directions, but only node 3 will have an action potential, not node 1. Node 1 will not have an action potential now because it is in refractory. So its sodium gates are locked shut and you can't have an action potential there. This continues. So now, while node 3 is having an action potential, you've got your potassium coming out, then you have your extra potassium coming out, and then this one goes into refractory. So while node 3 is having action potential and sodium is diffusing in all directions, node 2 is having refractory. So this means that the action potential in node 3 can stimulate node 4 to have an action potential, but not node 2. And this process continues down the whole length of the axon. And that's how you ensure that the action potential is moving in the right direction. So the action potential will only move this way. It can't go backwards, because every time a node has an action potential, that's followed by a refractory period, and that node cannot have another action potential yet. By the time refractory ends, the action potential has already passed on. So once we get to where the action potential is in node 3, refractory is done here. This is back to resting membrane potential. But you're too far away for that sodium to diffuse all the way back to node 1 and stimulate another action potential. So refractory periods are very important at keeping the signal moving in the right direction. And they work because that hyperpolarized state does not allow sodium gates to open. This is also going to be important when we get to chapter 19 and learn about the heart. Refractory periods are very important in the heart, so you might want to make yourself a little note to review this video when it's time to learn about the heart. Now that you know all about the axon and how it has an action potential, let's look at the receptive segment, the dendrites and cell body. So remember, these are the parts that receive a signal from another neuron. And that signal is in the form of neurotransmitter. And we can just start abbreviating neurotransmitter NT. So the gates on the receptive segment are ligand regulated. So remember, that means that they open when neurotransmitter binds. If you also recall from that first picture of the neuron that we labeled, you have 3 sets of gates here. We have the sodium gate, and it lets sodium in, and when sodium comes in, you raise voltage. We have the potassium gate that lets potassium out, and when potassium goes out, voltage goes down. We also have a chloride gate. Chloride, notice, is a negative ion. Everything else we've been working with is positive ions. Chlorides are 1 anion. When the chloride gates open, chloride comes in. And chloride coming in, because it is negative, reduces the voltage. When these happen, we get what are called graded potentials. So all of this movement here in the receptive segment produces graded local potential. This is different from the action potential that we had in the axon. The graded part means that it's only small changes in voltage. Remember, with the action potential, we went from negative 75 to positive 30. Here, we're only going to change a few points. And local means that the change stays there in the receptive segment. It's not spreading down the whole length of the neuron. So an action potential is not local, it spreads. Graded potentials that are local do not spread. It's those voltage regulated gates of the axon that allow spreading. Here, because you have to have the neurotransmitter to open the gates, even if the voltage changes, that does not stimulate the neighbors to open. So the potentials stay local. There are 2 different types of graded local potentials that we have in the receptive segment. Those are EPSP and IPSP. So the first one here is EPSP. This stands for excitatory postsynaptic potential. These are the ones that raise voltage. And if you raise voltage, you are encouraging an action potential to occur. Here's how I think of it. Imagine you're really, really hungry and you go out for pizza. And think about a time when you're so hungry and that pizza arrives and it's super hot. It's like lava, right? The waitress brings this pizza fresh out of the oven. It's got all this cheese and sauce that are lava. You know you should wait and let the pizza cool, but you're so hungry you don't want to wait. So you take a big bite of this lava pizza. Now it's burning your mouth. Now you have to decide. Do you just chew the pizza up normally and suffer through the pain? Or do you do that thing where you look like a fool and you like try to pull air in and out of your mouth without spitting the pizza out and cool the pizza off while it's in your mouth? And you look kind of like a monkey while you're doing it, but at least it relieves the pain. So you have to pick one or the other. Part of you wants to pull the air in and out of your mouth and not care that you look like a monkey and you're making a fool of yourself. And part of you wants to just suffer through the pain and chew the pizza up and swallow it. That's how it is with your neurons. Notice the receptive segment here. It is getting signals from several other neurons. And it can get conflicting signals. Some of them want it to have an action potential. Some want it to not have an action potential. So just like part of you wants to just chew the pizza up, part of you wants to pull air in and out of your mouth like a monkey and cool it off. You get conflicting signals. The EPSP is the signal that wants the neuron to have an action potential. So if you're going to have an action potential, you have to raise voltage. So you're going to let sodium in. So EPSP, you secrete a neurotransmitter that binds to the sodium gates. So this is an excitatory neurotransmitter. These green dots here are excitatory neurotransmitter. So to have an EPSP, you use excitatory neurotransmitter. It binds to the sodium gates, which causes them to open. Sodium comes in and voltage goes up. Notice here on the graph, here's our -75, here's -55. We just get a little blip there. Remember, these are graded potentials. So the voltage just goes up a little. The other type is inhibitory. So this is IPSP. This is when you do not want an action potential to occur. And to avoid an action potential, you reduce voltage. So IPSP reduces voltage to prevent having an action potential. So in this case, these neurons, you got 3 of them here, these 3 neurons do not want the next neuron to have an action potential. They're telling it to be quiet. So they want to reduce the voltage. So they are going to secrete inhibitory neurotransmitter. So these red dots are inhibitory neurotransmitter. And you can see by the color coding of the gates that inhibitory neurotransmitter is going to open the potassium gates and the chloride gates. So to have an IPSP, you have inhibitory neurotransmitter. This binds to the potassium gates and the chloride gates. When you open the potassium gates, potassium goes out. When you open chloride gates, chloride goes in. Both of these result in reduced voltage. And here you can see, here's our -75, here's our -55. It's a graded potential, so it's just a tiny little dip there in the voltage. But you're driving it away from having an action potential. So IPSP, it's telling the neuron, have an action potential, send this signal. EPSP is telling the neuron, be quiet, don't send any action potentials. A neuron can be receiving both types of signals at the same time. So if we look here, you can see the red is the inhibitory and the green is the excitatory. So this is normal. The neuron is always receiving both types of signals. So this is where the hillock comes in. Remember, we had the hillock is the initial segment. It's what decides which way to go. And that process of making the decision is summation. So think of sum is when you add numbers up. The hillock is basically adding up all those local potentials. The hillock has voltage regulated gates. And remember, voltage regulated gates open if you can reach at least -55. -55 is your threshold. So you start at resting membrane potential of -75. Then you have your IPSPs and your EPSPs. So you can see here you have, this one would be an EPSP. Here's another EPSP. Here's an EPSP. Here's your eyes, where the voltage is going down. The hillock adds all those up. So you start at -75. You have your local potentials occur. The EPSPs raise the voltage. The IPSPs lower the voltage, and the HILIC sums it up. If you can raise it by at least 20 points, then you will open the gates in the HILIC and have an action potential. So that means that EPSP has to win. Think of it like a tug of war. EPSP is raising voltage, IPSP is lowering voltage. If EPSP wins by 20, then you have an action potential. So if you look at summation, here you have a small rise, a small rise. They add up to make that bigger rise. Here you have a small decrease, a small decrease. They add up to make an even bigger decrease. Here you have a small increase plus a small decrease, and they cancel out. You have no change because they canceled each other out. That's the idea of summation. And there's two types of summation that can occur. We have spatial and temporal. Here you can see, here's an actual photograph of all these are the axon knobs. And here is that receptive segment. So you can see what it looks like in real life with how many other neurons are secreting neurotransmitter onto this one. So summation comes in two types, spatial and temporal. Spatial summation, where basically each neuron gets one vote. So you can see here, you have all these neurons. Some of them are doing the red inhibitory, some are doing the green excitatory, and they're each getting one vote. In temporal, some neurons get to vote more than once. So you can see here, you have the two rises. Notice on the graphs up here for spatial, there was just one rise in each box. Here you have two rises in each box, and they build on each other. Down here, you have two dips in the box, and they build on each other. So temporal is one neuron voting multiple times. I kind of think of this like nagging. You have one neuron that gets its way because it just keeps nagging or whining. It keeps secreting its neurotransmitter over and over. Okay, so if you can get the voltage of the receptive segment up to at least negative 55, then your voltage gates here in the HILIC will be stimulated to open, and then they have an action potential, and then because here you have more voltage gates, you get that process going, where then the action potential continues through the axon. One more topic in this video is to give you a couple of real-life examples of how we can manipulate the neurons to do what we want. So lidocaine. If you've ever had stitches, you've probably had lidocaine. That's what they inject in there to numb the area up before doing a medical procedure. So the whole idea of lidocaine is that it stops you from feeling the pain. Okay, so like this is what they use to numb an area before a medical procedure, such as getting stitches or having a mole removed. So the pain still occurs, but you are not aware of the pain because your neurons can't tell your brain. And if your neurons can't send the signal to the brain, you don't feel the pain. So lidocaine works by blocking the sodium gates. Sodium can't get into the neuron. And if sodium can't get in, you can't raise the voltage. So you can't have an action potential. So here we have a medicine that works by blocking one of these gates. Lidocaine blocks your sodium gates. Sodium can't get in, you can't raise the voltage, no action potential. There's still damage and pain occurring at that body part, but you can't feel it. The other one is anesthesia. This is what puts you to sleep for surgery. So again, you want to stop the neurons from being able to signal. This works by opening the chloride gates. So then chloride goes in and you reduce the voltage. So you hyperpolarize the neuron. So now because it's hyperpolarized, you're not going to reach that negative 55 and have an action potential. So you drive the neuron away from negative 55. And this is how you prevent the action potential. And then you are safely knocked out and they can do surgery. Well, we finally made it to the transmissive segment, the last part of the neuron. We're ready to see how this neuron communicates with another cell. So this whole picture that we're looking at is a synapse. A synapse is where the neuron is going to communicate with either another neuron or the effector. And it does so by secreting neurotransmitter. So let's start by labeling some parts here. This area right here on your synaptic knob is the presynaptic membrane. So remember, this whole structure is the synaptic knob. We labeled those when we first labeled the neuron. And its membrane is called the presynaptic membrane. And one of the important features of this membrane is that it has a negative charge. And you'll see in a few minutes why that is important. Then over here, we have our postsynaptic membrane. This is the membrane of either the next neuron's receptive segment or your effector. Remember, effector is the organ that we're telling to do something. So this could be a muscle or a gland. And it could be skeletal, smooth, or cardiac muscle, any of the three. Then you have a gap between them here. This gap between them is called the synaptic cleft. So this whole area between the two membranes is the synaptic cleft. So pre means before and post means after. So the presynaptic membrane is before the synaptic cleft and the postsynaptic membrane is after the synaptic cleft. So information is moving this direction. Remember, information always moves one direction. So the basic idea, we'll do the basic idea first, then we'll get into a little more detail. The basic idea is that the synaptic knob secretes neurotransmitter. So that is the red dots here. These little red triangles are the neurotransmitter. The neurotransmitter diffuses across the cleft and then it binds to receptors. So these blue things here are ligand gates. So it binds to receptors on the postsynaptic membrane. And the receptors are part of the ligand gates. And then that makes these gates open. And when they do, ions move in and out. And then you have the signal. Signal is received. Okay, now we can look at a little more detail of how this happens. The more detail is in the form of control. How do you control when you release neurotransmitter and when you don't? The neurotransmitter, notice, is kept in vesicles. So these are vesicles that contain the neurotransmitter. Also remember that the receptive segment has voltage-regulated calcium gates. And that when they open, calcium moves in. These vesicles have a negative charge. So remember when I said that it would be important that the presynaptic membrane has a negative charge? This is where that comes in. The vesicles and the presynaptic membrane both have a negative charge. So remember, like charges, they repel, right? Opposites attract, likes repel. So the vesicles do not want to get near the presynaptic membrane. If they do, they're pushed away by that electrostatic charge. So as long as they're pushed away, they cannot secrete their neurotransmitter. So here, anytime if this vesicle were to get too close to the membrane, it would be repelled back away from the membrane. When you do want to release the neurotransmitter, so you've had an action potential, so these lines, these orange arrows, represent an action potential. If you're having an action potential here, that means you're raising the voltage. So that means up here in your axon, you're doing that whole sodium in, raise the voltage. And then remember, the sodium diffuses. And when it does, it also raises the voltage of nearby areas. So when the action potential gets down to the end of the axon, you have an increase in voltage right here. These calcium gates are voltage regulated. So that makes these calcium gates open. So when there is an action potential in the axon, it raises the voltage. This then stimulates the calcium gates in the knob to open. The calcium comes in. And when calcium comes in, it coats those vesicles. So here you can see, here's a vesicle coated in calcium. Calcium has a positive charge. So when it coats the vesicle, it makes it positive. Now that it's positive, it is attracted to the presynaptic membrane because opposites attract. So now the vesicle will fuse with the presynaptic membrane and secrete the neurotransmitter. And if you remember from back in chapter 4, that is exocytosis. So now because opposites attract, the vesicle and the presynaptic membrane are attracted to each other and they will do exocytosis. And now that the neurotransmitter is released, It will diffuse across the cleft. It will bind to the ligand-regulated gates on the postsynaptic membrane. Then you'll get ions moving in and out here, and you will get a signal on the next cell. So that is how you transmit the signal from one cell to another. Now we have to look at what happens after the signal has been transmitted. You can't just leave that neurotransmitter in the cleft. So after the signal is transmitted, you have to clean up the neurotransmitter from the cleft. There's two ways to clean up that neurotransmitter from the cleft. This way is showing reuptake. So this is where you have transport proteins that will move the neurotransmitter back into the knob. The other method is down here, number 2. You can have an enzyme that breaks down the neurotransmitter. So right here, acetylcholine, ACH, is our neurotransmitter. So first it's released by exocytosis right here. It diffuses, it binds so that the signal is received. Then it comes over here and this enzyme breaks it. Here's the little broken parts. Then the broken parts do go back into the knob. But before they do, they're broken by an enzyme. So either way, you have to get that neurotransmitter cleaned up. You have to deactivate it so that it can't keep repeating the signal. So the purpose of this is you want to avoid repeating the signal. So that is why you will either take up or break down the neurotransmitter. Okay, then we do have neuromodulation. So neuromodulation is where you don't necessarily clean up the neurotransmitter right away. So in neuromodulation, you're going to alter what happens with the neurotransmitter in the cleft. Okay, and a lot of times you're going to act to keep the neurotransmitter in there so that you do repeat the signal. So an example of this is cocaine. You can see in this picture, cocaine has been added. Cocaine are these red things here. Here is that transport pump that will do reuptake of dopamine. Cocaine, notice, blocks this. So if you take cocaine, here's the normal situation. You release dopamine, it binds, then it goes back into here. So normal, here's the dopamine is released, it binds to the receptor, then it goes back, and you get that feel-good signal once. Right? Dopamine transmits a signal of feeling good. If you have cocaine, you release the dopamine, it binds, you get that feel-good signal. And then when the dopamine goes to go back, its way is blocked. It can't get back in to the synaptic knob. So then it bounces back and it binds again. So then you get another feel-good signal. And this just keeps repeating over and over. Every time the dopamine tries to get back into the cleft, the cocaine is there blocking its way back in, so it bounces back, binds to the receptor again. And then you repeat the signal. Another example of neuromodulation is with botulinum toxin. So let's review what goes on with a muscle. And we're going to put some ideas together from Chapter 10 and Chapter 12. So this picture should be familiar. Remember, the whole idea is that you get an action potential on your sarcolemma. That goes down the T-tubule. And when it does, you release calcium from the terminal cisternae. And that calcium, here's all that calcium. Remember, the calcium binds troponin. And that moves tropomyosin. And then you get cross bridge and power stroke, and the muscle contracts. Okay, so let's tie in some stuff from Chapter 12 now. Here is your neuron that's having the action potential. So we would have an action potential on this motor neuron. We get down here. Remember, we're going to let calcium in. It's going to make the vesicles turn positive. They're going to fuse with that presynaptic membrane. You get exocytosis. And in this case, that neurotransmitter that we're secreting is acetylcholine. Acetylcholine is the neurotransmitter that is used to stimulate skeletal muscles. The acetylcholine diffuses across the cleft. Here are the receptors on our postsynaptic membrane, and now our postsynaptic membrane is the sarcolemma of a muscle fiber. These gates open, they let sodium in, potassium out, and that creates an action potential on the sarcolemma. Then when you get over here, that action potential goes down the T-tubule. The terminal cisternae has voltage-regulated calcium gates. The terminal cisternae is just like the synaptic knob. The terminal cisternae has voltage calcium gates that open when there is an action potential in the axon. In the T-tubule. Just like the synaptic knob will open its voltage-regulated calcium gates when there is an action potential in the axon. So there's a lot of parallels here. You have an action potential in the axon, makes calcium gates in the knob open, and you release neurotransmitter. Over on the muscle, you have an action potential on the sarcolemma and T-tubule, makes voltage gates in the terminal cisternae open, and you release calcium. So back to the botulism and how this botulism is going to affect this process of the neuron stimulating the muscle. So let's think about botulism. It has become a lot more rare. But if you think about older people you've been around, maybe your great-grandparents, or if you work in a nursing home, do you ever notice they have this thing about dented cans? They don't want to eat food from dented cans. The reason for that is before we got better technology like we have today, you could get botulism in your canned food, and it was a type of food poisoning. So botulism, first of all, it comes from a bacteria, and this bacteria tends to want to grow in canned foods. Now, today, technology and sanitation have improved, and it's very rare you're going to go to the grocery store and get some canned food that has botulism in it. But years ago, before the technology was what it is today, you did sometimes get botulism in canned foods. And when the botulism grows in the canned food, it makes bubbles. And those bubbles can dent the can. And that's why people who are old enough to remember when you did need to worry about botulism don't like dented cans. It's a sign that it has botulism. So anyway, what happens is the bacteria makes a toxin. And this toxin from the bacteria gets into your synaptic cleft. And it blocks acetylcholine. So you cannot have the acetylcholine to stimulate your muscle. So back up here, where you're secreting acetylcholine, and that acetylcholine is binding to the gates on the muscle, that does not happen with botulism. So if you had botulism, you would not have this. So botulism causes flaccid paralysis. Your muscles can't contract. And this can be deadly because then you stop breathing. So no, your heart wouldn't stop beating. This affects skeletal muscle, not cardiac muscle, but you rely on skeletal muscles to breathe. So although it's rare today to get botulism from your food, some people purposely inject themselves with it. That is the whole idea behind the Botox injections. It's a little bit of botulinum toxin, and the doctor injects that into a muscle and it paralyzes the muscle. And people do this. It can be used like to stop the wrinkles, because if you can't move the muscles on your face, you can't wrinkle your skin. It's also used for people who are having really painful muscle spasms. They can inject a little bit of the Botox in there and stop the muscle spasm and the pain.